Hi, I'm Irina Stepanov. I'm Mayo Professor in Public Health with my research program based at the Masonic Cancer Center at the University of Minnesota. I'm really excited to give you this virtual tour of our lab and tell you about our research program. So one of the key areas in my lab is examining the harmful potential of tobacco products by analyzing the chemical composition of these products and better understanding what are those toxic chemicals and carcinogens that might lead to the development of disease in tobacco users. We use this information to investigate the public health impact of different products and to develop regulatory measures and also to inform consumers and healthcare professionals. Another big area in our lab is development and application of biomarkers to understand susceptibility to disease in tobacco users. As you know, some people and even some population subgroups are particularly vulnerable to the effects of tobacco use. So we are developing and applying novel biomarkers to better understand the mechanisms that underlie these disparities and lead to the development of tobacco-associated diseases. Our third important direction in our group is developing capacity for global cancer prevention. As you know, cancer is a global problem, and tobacco use is a major preventable driver of risk. So through our work, we inform global tobacco control by developing capacity for research on tobacco carcinogen exposures. The knowledge and the resources that we are developing through this work can be also leveraged to advance cancer prevention in other areas. So now I will take you to the lab to show you how exactly we conduct this research. Our lab is located in a beautiful cancer and cardiovascular research building on the University of Minnesota campus. There are many researchers working in the laboratories here, and they use cutting-edge technologies to solve a really wide range of cancer research challenges, from mechanisms of cancer all the way to drug development. Okay, so we are here on the second floor of CCRB, where all the laboratory magic happens, where we do this work that I outlined earlier on tobacco analysis and biomarker analysis to understand the impact of tobacco products on human health. One part of analysis is looking at tobacco products themselves. So some of the tobacco work is being done here in tobacco room, just to keep the potential contamination contained and not expose some biological samples to tobacco chemicals. And in this room, what we usually do, we weigh tobacco samples that we then place in tubes and extract with different solvents. So we have dedicated bench space and dedicated analytical balance for this purpose. But we also condition cigarettes before smoking them on the smoking machine. And we use this temperature and humidity control chamber for this purpose. For example, about 2 days before we need to smoke cigarettes, we place them here in this chamber. And you can see here some tubes with cigarettes that are being prepared to be smoked on a smoking machine. So this is an important step in cigarette analysis. And after this is done, the cigarettes are taken to the main lab and smoked on the smoking machine. And this work is done in a fume hood to protect researchers from toxic exposures to cigarette smoke or e-cigarette aerosol. So this work happens here using this smoking machine. This is Alexandra Alcheva. She is a PhD student in my group. Alexandra has been involved in several studies in which she analyzed tobacco products. Historically, our laboratory was focused on analyzing traditional products such as cigarettes and smokeless tobacco, and even though such products have been on the market for a long time, there are still very important gaps in understanding the composition of these products, and we still generate important new information that can inform public health efforts. For example, Alexandra recently analyzed smoke from natural American spirit cigarettes, which are marketed as organic and additive-free, and she showed that, in fact, such cigarettes exposed smokers to higher levels of nicotine than many other brands. When we smoke cigarettes in the machine, we use special filters to trap smoke particles so that we can later extract different chemicals and analyze them by mass spectrometry. We first program the machine to tell it how we want the smoking to proceed. For example, we tell the machine how long each puff needs to be and the interval between puffs. We then put the holder with the filter pad in the machine, set the cigarette in, light it up, and start smoking. The sensor tells the machine when to stop smoking the cigarette. When smoking is done, we remove the filter from the holder. You can see here how much tar is generated by just one cigarette, and with each cigarette this adds up. For example, this is how much tar goes into the lungs of a 10 cigarette per day smoker over 1 year. There are thousands of chemicals in cigarette smoke, and we are focusing on the most harmful ones, particularly those that cause cancer. We save each filter pad separately, freeze it, and later extract it with solvents, purify, and analyze by mass spectrometry. In addition to traditional tobacco products, our lab has been working on characterizing electronic cigarettes and other new types of tobacco products. These products have been on the market for a relatively short period of time, and there are still many unanswered questions about the potential impact on public health. For example, for smokers switching from regular cigarettes to electronic cigarettes, exposures to many harmful chemicals may be reduced, but long-term effects of e-cigarette use is not clear. In addition, there are many e-cigarette users who never smoked, especially among younger people, and it is important to know what such users are exposed to and the consequences of such exposures. There is no burning process when e-cigarettes are used, but there are still many harmful chemicals that are formed when the liquid is heated during puffs. Such chemicals are primarily volatile gases, for example, formaldehyde. To trap these chemicals, we use these glass impingers filled with a liquid that can convert these unstable chemicals into more stable compounds, so-called derivatives. And we analyze these derivatives by liquid chromatography. Okay, so after tobacco product analysis is completed and we know exactly what kind of chemicals are present there in what amounts, we need to analyze biomarkers of exposure to these chemicals in people who use tobacco products. And we do this work in a separate space to make sure that there is no cross-contamination of biological samples from chemicals present in tobacco products that was done in the other hood. So this is where we have a dedicated space, a dedicated hood for the analysis of biological samples. This is Katrina Yershova. She has been with my lab for over 10 years. She has done a lot of work on the analysis of tobacco products and more recently started working on biomarkers. We usually work with urine and blood, and this requires many purification steps to remove proteins and other molecules that can interfere with the analysis of biomarkers that we are interested in. A typical purification step is so-called solid phase extraction, which is purification on special cartridges, and you can see Katrina doing such purification now. The cartridges are first conditioned with methanol, then we use a positive pressure device which pushes nitrogen on the cartridges to help the methanol pass through. When this is done, we apply samples. Each sample goes into its own cartridge to be purified. And to avoid cross-contamination, we use a new clean pipette for each sample. When samples are absorbed, we add solvent that helps to keep biomarkers on the cartridge and wash off impurities. We use the nitrogen device again and repeat this several times before extracting the biomarkers from the cartridges and analyzing them by mass spectrometry. And lastly, when we want to better understand what these harmful exposures actually do to macromolecules, for example, to DNA, then we conduct this work on analysis of DNA adducts, which are DNA modifications caused by tobacco chemicals and other harmful chemicals. And this is, again, we need to have a dedicated space for this type of analysis. This is where the analysis of DNA adducts happens. This is Chris Ruschek. He has been with my lab for more than 5 years. Chris is expert in DNA analysis, which is very complicated and requires special skills. We usually use blood as the source of DNA, and we need to store blood samples at very low temperatures, minus 80 degrees Celsius, which is approximately minus 112 degrees Fahrenheit. This is an example of capillary tube with a blood sample. It needs to be handled very carefully, and because the temperatures are so low, you can see Chris wearing a special glove and using forceps to handle the sample. When we analyze DNA adducts, our first step is to isolate DNA from the blood. This is done by using different enzymes that we add to each sample, and these enzymes break down cells and remove proteins. As you can see, the enzymes are kept on ice because these are very sensitive processes and we are trying not to lose any DNA. Also, similar to the work with urine, we are very careful to avoid cross-contamination of samples. So we are using a new pipette tip when we add enzymes to each sample. After the DNA is isolated, we use purification steps that are similar to what you saw for urine purification. And when samples are ready, we analyze them by mass spectrometry. We are using the Masonic Cancer Center's mass spectrometry shared facility right here in the same building. And this facility is one of the best academic mass spectrometry labs in the country. So we are very fortunate to have access to this resource. There are many instruments here, from the very robust ones that can be used for analysis of tobacco products to very sensitive equipment that can be used for extremely precise and selective analysis of trace levels of biomarkers. A typical instrument is composed of a system of pumps that push samples through the chromatographic columns. You can see the very thin metal tube here. And there is also an autosampler and a mass spectrometry unit that detects chemical compounds present in the sample. The instrument is controlled by a computer running specialized methods we developed for the analysis of different tobacco constituents or biomarkers. When we have our samples purified and ready for analysis, we set them in the autosampler, and then we initiate the computer program that tells the instrument to start working on the first sample. Autosampler draws the sample from the vial, injects it into the mass spectrometer, and then rinses the needle before the next sample is injected. We can see the results as soon as each sample is analyzed. We look at the signals produced by the instrument, and the magnitude of the signal allows us to determine the amount of a constituent or a biomarker that was present in each sample. These data, once collected and analyzed, are published in scientific journals and presented at conferences to share our findings with other researchers, with public health professionals, and regulators. Thank you for watching this video and learning about research program in my group. I hope you enjoyed the virtual lab tour and learning about all the methods and equipment and exciting work that we do in the lab. Please do not hesitate to reach out to me if you have any questions, if you want to learn more about our work, or maybe arrange an in-person tour in the lab. I hope that will become possible soon, and I'm really, really looking forward to this. Thank you.